Hi everyone, welcome to HSC Trial Revision 8 Notes lectures. My name is Aditi and today I'll be taking you through the HSC Biology lecture. I hope we are all doing really well and having a lovely start to the holidays. So a very warm welcome to everyone. Now before I get started, I'd just quickly talk you through some of the amazing resources that we have to offer here at 8 Notes. You can find lots of downloadable study notes on our website. We obviously have lectures going on this week, so try and attend all the lectures that are relevant to you. There's no limits on how many you can attend, and you also have past lectures available to you as well. There is also an amazing discussion forum online, where obviously we are on the forum and so are like lots of other students and past tutors as well, and it's a lovely community that again wants everyone to succeed. We also have online videos, newsletters, ADA calculators that will be helpful later down the track, and also different articles which include study tips and strategies to help you smash out the year. And like I said, you can find them all at atanotes.com. Along with that, we also have some more resources. So if you're looking for that extra push, we've got TuteSmart, which is our online tutoring resource from recent year 12 grads. We also have study guides that are available via our 8 Notes shop. They are printed revision materials that are amazingly set out, might I add, and a wonderful resource to help you get all your notes in one place, really easy to use. And I say that because I've used it. And along with that, complementing that, we have Ed Unlimited, which is kind of like a Netflix for all of our study guides. So you've got study guides, topic test booklets. For English, you've got your text guides as well. And again, having used them, amazing resource. So if you are again looking for that bit of extra push, that extra help, definitely check them out. So let's get into it today. Like I said, we are going to be revisioning for HSC trials, and the purpose of this lecture is to help you feel confident, and to remove any doubts that you may have regarding any of the content points. Now today's lecture will be based on a module 7 review and then a module 8 crash course head start if you haven't done module 8 yet in school. And please feel free to ask any questions that you may have. I'll be live on the chat, so ask any questions, even if they're from mod 5, 6, or general questions about studying, whatever it is. I'm here to help you with anything today. So yeah, today you're kind of trying to join all the dots and make everything sort of come together, and to feel confident in being able to consolidate all of these different topic points across the modules. So a bit about myself. My name is Aditi and I graduated in 2021 as Dux with an ADA of 95, and the subjects that I've done is English Advanced, Extension, PDHP and Biology. I've also done Maths 3 units, Advanced and Extension, and then Chemistry. But I always like to say we don't talk about Maths 3 unit and Chemistry in here. But if you have any questions regarding that, or my experiences of it, or study advice or whatever, feel free to ask away. I'm currently at the University of Sydney studying a Bachelor of Science and Bachelor of Advanced Studies in Medical Science. It's a double degree, and I'm majoring in medical science and political economy as a Delhial scholar. Generally love reading, good shows and sunny days. Huge Harry Potter fan and a big classics fan as well. That little image over there is my little collection of all the classics so far. We're trying to build up on it. Well, not all of them are classics, but like this bit there, that's my classic section, and that's some amazing other books that are up there in my reading list. So let's get started with today's content. However, before we do that, let's have a look at where we are. Right, you are we're in July right now. The exam is in October, specifically on the 23rd of October. Um, so I'm imagining that right now we're all pretty stressed in terms of the trials coming up, and bio is obviously a very, very content heavy subject. So there's certainly that element of nervousness and anxiety in, having to remember everything. But I think my biggest tip with biology is focus on understanding rather than remembering, and that can go a long way, I think. So right now I would say start off by having a really good understanding of the syllabus and also where you are in regards to the syllabus. Do you feel really confident with some elements, or do you feel that some parts of the syllabus are like, yeah, no, I don't want that in the paper? So whatever it is, like sort of sitting down and thinking where we're at with things is really important. And I think a good way to do that is to use like the traffic light system, which goes, you highlight things red, yellow, or green. Red obviously stands for things that you don't want in the paper. Like, nah, if it came, I would have no idea what to write. So you highlight things that you don't get, that you don't understand, as red. Then you go to yellow, where it's like, okay, I can write some things on it. If it's like a 4-marker, for example, I might be able to scrape 2 marks or 1 mark, right? And that would be your yellow, where you're kind of like, okay, I'm kind of getting that. And then your green, things that you would highlight green would be the ones that you're like, I can smash this topic. Like, I know everything and I am going to get 4 marks. So, and also, like, be kind to yourself with this. Don't think that everything's going to be yellow or red. Because when I tried this method, I looked at the paper, everything was yellow and red. And I was like, not really. You know what? I could probably have like do like all the questions regarding this topic, so I don't have to highlight a yellow. I can actually take go with green, highlight a green. So I think you can sometimes feel like, I don't know, you're being a bit overconfident if you highlight things green, but don't think of it that way, and that's from experience. Try and just go, okay, I will be able to answer most of this, and that's a green for you. You would always do better than you expect yourself to do. I think that's a good thing, a good motto to have in your head as you go through it. So yeah, have a good understanding of the syllabus and where you are in the syllabus. In prep for the trials, revise and memorize content. Complete half of the NESA practice paper. So this is basically so that you can, use 2 papers now and then 2 in the lead up to the HSC. You can obviously find lots of trial practice papers from other schools online as well, like past papers as well. So you can definitely check that out. But right now, it's about setting up that foundation for the trials and then ultimately for the HSC. So study notes also is something important to think about. Draft one of your first 3 module notes close to finished. Begin to condense and revise them, and you can always do that in tandem with the practice papers, and they can be refined as, like I said, as you put them in your practice papers and complete your practice papers. Now, the second stage is when your trials begin, right? And we all feel like that cat there, where it's like, it's certainly a journey. So we totally understand the pressure is there, but I think in a way it sort of definitely helps you feel confident for the HSC itself, because you've done, it's like a practice run. And I know, like, sure, it's worth usually 25% or something in your grades in class, so like in terms of the assignments, but nonetheless, remember, your 50% of your HSC, like 50% of your overall mark, is still locked away until you get to the HSC. It's in that treasure chest and you have the key to open it. So trust yourself. So trials begin, and then finally, as we get closer to the 23rd, we want to complete all the we want to complete all the NESA papers. We also want to make sure that our notes are up to scratch and that we are feeling confident that there's no reds in that syllabus outline anymore. Most of it should be green. There could be a couple of yellows, but that's the thing you'll be sort of sorting out before a week or so before the exam. You're like, okay, this is yellow, let me go back and have a look at the the content, like the dot point, and sort that out. So that's sort of why I would say, are like the 3 big milestones as we come closer to the 23rd. Okay, now I'm gonna try and make sure that I stick on time, just because we do, like I said, have a lot to get through today. There may be parts based on how I feel, like, from how I feel, like, if they'll be pretty well understood by you guys, or parts that I definitely know like everyone needs revision on. So yeah, let's do that. So the syllabus, we've got module 5. We covered that in January. Well, we covered, like, we revised it in the January lectures, but we covered the whole thing in itself in October, in the October HSC lectures last year. Module 6, module 7, and then module 8. So module 7 was in April, and then module 6 we covered in the 8 notes lectures in January. So you can definitely check them out on our website. You have the past lectures available to you. And then obviously we're going to cover module 8 today. So let's revise the key concepts. Like I said, I'm going to try and go through it, smash it out. Just, and if I feel like there are bits where I feel like, we'll be okay, I'm going to skip over them and spend more time on parts where I know we need to spend more time. But obviously you'll have the slides to look at them. You'll have access to the slides and obviously to the recording of the lecture as well. So that's something I should have mentioned earlier, actually. You'll have access to the recording, to this recording, even after the live stream time has finished. You will find this recording on the exact page on which you are accessing the stream right now, and you'll also have the slides available to you as well. So let's get a start on it. Now, causes of infectious disease, how a disease is transmitted. So we here want to start off by classifying different pathogens that cause disease in plants and animals. So we've got prions. They are Britannia's infectious particles. So some examples are mad cow disease or Kuro, and they are basically the smallest types of pathogens, like really small, about I think 200 amino acids chain long, so it's very small. Now viruses are non-cellular. They do have nucleic acid and a protein envelope, and they replicate inside living cells. Examples include measles, AIDS. You can also think about SARS-CoV-2, and that would cause COVID-19. Measles causes rubella. So measles is caused by rubella. HIV causes AIDS, and SARS-CoV-19 is the pathogen that causes COVID-19. So bacteria is the next one. So this is in terms of like smallest to it's going up. So bacteria is the next biggest. They're single cell, they're prokaryotic, they reproduce by binary fission. So this kind of links back to like binary fission, links back to module 5, so you should know what the process is. And they secrete toxins, invade cells, and also form biofilms. So salmonella causes salmonellosis. Mycobacterium tuberculosis causes TB, tuberculosis. Then we've got protozoa. They are single cell, they're eukaryotic. Examples include Plasmodium protozoa, which causes malaria. We looked through malaria in this module in a couple of different dot points, so hopefully you'll be very familiar with it. But we also look at dengue, and I think dengue is another disease that we look at as well when it comes to like epidemiology and controlling the spread of disease and so on. Then we've got fungi, which is eukaryotic. It can be multicellular, and they spread by releasing pores. The Candida albicans causes thrush. And then finally we've got macroparasites. So they are visible to the naked eye. Ectoparasites are those that live on organisms, and endoparasites are those that live in organisms. And tapeworm causes the tapeworm disease. So those are the examples. So always know your examples. And obviously macroparasites are the biggest pathogens. So you want to remember their size. the characteristics of each, the method of replication, try and have a disease for each one, and try and pick diseases which cross over different areas of the syllabus. So for example, malaria is a really good example of that, because we kind of come back to, we really talk about malaria in module 7 across different dot points, and then think about how you would actually distinguish them from one another. Okay, so now let's look at the work of Robert Koch and Louis Pasteur. So Koch was a German microbiologist. He identified microbial origins of anthrax, cholera, and tuberculosis, and he developed a procedure for isolating and identifying microbes, so that to say that this is the microbe that is causing the disease, and he did that through his postulates, which link microbial growth as a causative agent for the disease. So essentially, his postulates included that firstly, in all organisms with a disease, there is something that is actually causing the disease, a microorganism that is causing the disease. And then he sort of goes on to say that the microorganism has to be isolated and then grown in pure culture, and from there we inoculate a healthy animal with that with the microbial that we grew in the pure culture, and that is to see if that animal develops the same symptoms as the original sick organism. And then from there we isolate and regrow that microorganism from the newly infected organism. So we do the same thing, so it's kind of like a cycle, so we repeat it twice, and if the same thing happens, we've done it 2 times, and we can see that each time it's causing the same symptoms, and it's obviously then due to that microorganism. We've found what microorganism it is that is causing the disease. That is what his the 4 postulates are doing. It's about identifying that this certain microbe is causing the disease. Now, Louis Pasteur, he was a French microbiologist. He disproved the theory of spontaneous generation, and he also proposed the germ theory of disease. And the germ theory of disease is essentially saying that all things are coming from pre-existing things. Like all of these, diseases are coming from things that are already in nature that pre-exist. It's not just happening out of nowhere. And that is kind of what the theory of spontaneous generation was, that it just, happens on its own. So how did he do that? Essentially, he conducted an experiment called the swan-necked flask experiment, and within that, what happens is that we take flasks with bent swan necks. So as you can see here, this is what we are referring to as swan necks, and essentially what we do is we heat it up, we let the flask sit, and we do it twice. So with the first set, we heat it up, we let the flask sit, and we don't do anything to the swan neck flask. We leave it as it is and just let it sit. In the second one, we heat it up, but then after doing so, we remove the neck and just let it sit on its own. And what we would find after that is that in the flask that we actually opened up, the bacteria would be present there, and that's telling us that microbial growth is happening because of the microbes in the environment. Here there's no way for the microbes to actually make their way through the flask. Sure, they would get in, but because of the structure of the neck here, it's hard for them to get through. However, here we leave it open, they're going to be in the flask, and that is telling us that something in the environment is that these microbes exist in the environment. That is what it's essentially telling us, and that's Pasteur's experiment. Amazing. Any questions? Again, feel free to ask away. No question is a silly question. Now let's have a look at the adaptations of different pathogens that facilitate the entry into and transmission between hosts. So entry. How do different pathogens actually enter the host? Firstly, we've got cell wall degrading enzymes, and as the name suggests, these enzymes, they break down the cell wall to release intracellular nutrients and make their way into the cell. Toxins, they damage host tissues and they disable the immune system, and that's the key here. By disabling the immune system, you're kind of getting that that free entry into the cell, and you'll be pretty safe. Example includes botulinum. Then adhesion to host cells. This is where adhesion molecules on the pathogen help the pathogen stick around cells, and then from there colonize tissues and organs as well. Effector proteins. So proteins that are secreted to suppress the host defenses and actually aid their entry into the cell. And something very similar happens with the COVID-19 virus, where it has effector proteins that ultimately hijack the machinery of our cells. They mess with the protein synthesis and so on in order to actually gain entry into the cells. And then finally, we've got extremophiles, and these are the pathogens that can survive, as we can see by the word extreme, in extreme environments, in hostile environments, to help their survival inside of organisms. So that's how they gain entry. Now, when we compare the transmission, we also need to think about essentially how is it happening. We know the internal chemical or biochemical formula of what the the different parts of these pathogens are actually doing to enter the cells, but how do they actually transmit from one to the other? So, protective coverings. Bacterial capsules, what they do is that they're tightly packed polysaccharides, and they are giving the bacteria an extra protective layer. And we also have viral envelopes, and they allow the longevity outside of the host cell and actually also help with the avoidance of the immune system. Then we've got vectors, and a big example of vectors is malaria. So malaria, the Plasmodium protozoa, which causes malaria, is carried inside mosquitoes. So essentially what vectors do is they increase the efficiency of transmission, as you can imagine, from a mosquito to a human, to another mosquito to another human. Like it's pretty fast spreading. So they carry pathogen from host to host, and in the case of malaria, it's not all mosquitoes actually spread it. It's a specific species called Anopheles mosquitoes, and that two female Anopheles mosquitoes actually cause the transmission of Plasmodium protozoa, which then causes malaria. Then reservoirs. So this is where sites for both living and non-living, where pathogens lay dormant for a long period of time between outbreaks, and animals actually can be the reservoirs of human disease, and big examples of that is rabies and cholera. And then finally, rapid species evolution. So this is when we've got high mutation rates during replication, which allow pathogens to rapidly evolve, and they increase the pathogenicity of the pathogen, obviously its potency, and then also increase its ability to actually get into the host cell and evoke that immune or rather evade the immune response. It's something like COVID-19. Obviously, we can, from the pandemic, we saw how fast it mutated. We went from alpha to beta to gamma to omicron. And obviously delta as well. So it mutates quite fast. Okay, so in summary, pathogens spread disease. Know an example of how disease is spread in terms of understanding the practical side of the module. You want to know about Koch's postulates. You want to know about Pasteur's experiment and what exactly it involved. Modes of transmission, we've got horizontal, vertical, Robert Koch's postulate, so that we've just spoken about, and the swan-necked flask experiment. Agricultural diseases are also very prominent in both, obviously, plants and animals. So know your causes and effects. Always good to have an Australian perspective, so you want to have examples of agricultural diseases as well. And finally, pathogenic adaptation. So how do actually pathogens gain entry into the host cells? So think about enzymes, proteins, toxins, and then transmission. How do they go from host to host? Protective coverings, reservoirs, vectors, and evolution as well. And that's the first stop point done. Okay, I think we're going pretty well according to time. So let's move on to responses to pathogens. So this is where we want to know how a plant or animal respond to infection. It's a pretty small dot point, and you just want to know an example of how does a plant or animal respond to the infection. And in the syllabus, you can choose whether it's a fungal pathogen you're looking at or a viral pathogen. So I'll leave that one out for today, and we'll move on to immunity, because this is the one that really I think you just need to have a really good understanding of immunity and how things are happening. So the inquiry question here is, how does the human immune system respond to exposure to a pathogen? So follow through this with me and please ask questions. Again, it's going to be like a summary of all things, but last series, the lecture series, I went through this in a lot of detail. So if you want that extra bit of detail, go make sure you check that out as well. But today we're kind of gonna go a bit summarized and quickly through it, and then we'll move on to module 8. So the first line of defense essentially includes cilia, which is the hair-like projections. They line airways and they move in wave-like motions to actually push the pathogens away. We've got skin. It's a protective layer. The pores secrete antimicrobials, and the outer layer is constantly shedding. We've got chemical barriers within our body. So stomach acid is one, alkali content of the stomach intestine, of the small intestine, and enzymes in the mouth as well. Then we've got mucous membranes. These are the cells that are lining the opening of the body, and they secrete mucus. And what they do is essentially trap pathogens and microbials in the mucus. And finally, secretions. Things like sweat, our sweat glands, hair follicles, urinary tract, they secrete fluids, and they do that in order to flush out pathogens, and some of them also secrete antimicrobials as well. And that's the first line. This is our natural, innate line, the first sort of line of defense that is always there to protect us against pathogens. Now, the second line of defense includes our lymph system. It also includes inflammation as part of the second line of defense. So imagine if you step on something sharp, something really sharp, let's say, I don't know, a needle or something, or a stick. For example, you're out in the park, step on a stick, obviously that stick's going to have a lot of bacteria on it if it's out in the park. Now, along with that, as soon as something harmful is going to enter the body, we're immediately going to see inflammation. Immediately, we're going to see swelling and redness, blood flow to the area in order to actually deal with that. And this process is clearly very it's an innate process. It's same for everything. Phagocytosis is also another example. So this is where specific cells, they see a bacteria and they engulf the bacteria, and then using lysosomes they dissolve and kill the bacteria off and then secrete it out. That's phagocytosis. Finally, cell death to seal off pathogens. Now this is apoptosis, and what happens is essentially this is a normal cell, and if it is infected, if something has hijacked the cell, what we do is there are signals that ultimately tell the cell that you have to die because you are infected. We can't have that spreading to other cells around you, so it is going to start condensing and then fragment and break off, and that's apoptosis. That's cell death. Now, what is the difference between the two? Essentially, the innate immune system includes both the first and second line of defense, and it is always there. It is always protecting us. It doesn't, appear or doesn't sort of start working when we are hurt or when something happens. It's always there. Now, it provides us innate protection against pathogens, and it is non-specific. So it's not going to react differently to different pathogens. It's going to react in the same way to all pathogens. It's quite a generic way to react to all of them. The response rate is pretty fast. Like, as soon as, again, if you step on something really sharp in the park, branch or stick or whatever, the response is going to be very, very fast. Doesn't need time to develop, and there's no immunological memory, which is why it responds to everything in the same generic way. Now we will look at adaptive immunity, but before we do that, just consider that firstly, it's not always there. So what happens is that it gives us adaptive protection against pathogens. The immune response essentially here is giving us adaptive protection against pathogens. It is a highly specific defense, so it's going to it's going to react differently to all different pathogens. There is specialized protection against certain pathogens as well. Slower response rate though, because it needs that time to actually determine what it's going to do, how it's going to do it. You're going to find that out in 1 second when I go through it, how it's actually happening. And it needs time to develop, and there is immunological memory. So think about, for example, the flu, the influenza every year. If you've got the cold, whatever, we'll see that obviously the influenza mutates and so on, but we would see that the immune response is actually pretty fast to that, in comparison to something like COVID-19, where the response rate is different because our system hasn't actually we haven't dealt with that before. Well, most of us haven't. So that's the main difference between innate and adaptive immunity. So how does the adaptive immunity actually work? Essentially, think of it this way. Firstly, we've got an antigen. Now, the antigen is present on the cell surface, on the surface of the pathogen. And we want to create antibodies to the antigen so that we can send out antibodies in our body, and we can use that to actually identify and to capture the antigen. And each antigen is going to be different, so it's going to have a specific antibody. And this is where the idea of this process taking time comes in. It's not immediate. It takes time, especially if it's obviously when it's a new pathogen that the body is facing. It is going to take time to actually determine what the antibody is first, and it's a lock and key model. So it's a lock and key model, antigen antibody interactions, and antibodies are the molecules which recognize the antigen. Antigens are the cellular markers that are found on the surface of the pathogen, and it's a lock and key. So one of these antibodies is going to fit into that antigen, and that is going to help the cells, the immune cells, actually recognize that, hey, that is the antigen, that is what we need to kill. So how does it happen? I'm gonna explain you the whole concept via an analogy. So think of it this way. I just moved into this body, but I'm all alone. We've got a pathogen here, right? The pathogen, and it has an antigen on it. That's the first thing we need to remember. Now, this is where we go through a concept called clonal selection. And clonal selection is essentially when, let's see, we put this is where we kind of need to have a match between the antibody and the antigen. And again, the analogy here is, for example, being on a dating app, right? So we're going to search for a match. This is our pathogen, right? We're going to search for a match. And the concept is clonal selection. So we've got the first match. Yeah, it's not matching, right? Antibody one doesn't work. So let's say it's a Y shape. It's not working. So we move on to the next one. Another antibody, not going to work. We move on to the next one. Still not working. Move on to the next one. Then we find the antibody. Okay, we found antibody, and here it is. It's fitting perfectly, and essentially it's gonna fit each other, and that is going to be the antibody for the specific antigen. So that analogy was just to help you understand that it's a process. We're not going to immediately find the right antibody. It's going to take time, and we need to find the right one, and that is why, again, the process takes time to develop. So what is happening now, and the bigger picture of things, is this. So we've got a pathogen, right? So we've got a pathogen. Firstly, it is going to be engulfed by a macrophage. Now, a macrophage is going to have something called the MHC receptors, and essentially it is going to display the antigen of the pathogen on its surface. Now, when it displays the antigen displayed on the markers like that, so that's the antigen, it's displaying the antigen on the surface. It is then going to we're then going to have interleukins called helper T and B cells to the site of infection. So remember, we've got B cells versus T cells, and the helper T cells, firstly, they're going to come up. They've got the antibody for the antigen, right? And they are going to call 3 other cells. Now, the helper cells, I like to think of them as the kind friends that are going to be always there for you. So as soon as the macrophage is like, hey, I've got an antigen, helper T cells are going to come. Then the helper T cells are going to call the rest of the group. Oops. The first one is the memory T cell. Now, the memory T cell is the, I guess, the friend of the group that's like, okay, we need to make a list. We need to remember this, the really organized kind. And it's going to remember what antibody we actually used for the antigen. So this is where, as the name suggests, we're going to memorize that. We're going to remember it. This is where immunological memory comes in. Then we've got suppressor T cells. No, firstly, let's go through killer T cells. Now, killer T cells are your friends that are going to be like, the really aggressive types, the ones that are like, yeah, I don't care, let's do it. So these are the ones that are actually going to go ahead and secrete perforin to actually destroy the pathogens, and they can go rampant, like they can really go rampant. So these are really the really cool, oh well, yeah, they're always the aggressive types, the ones that are really quickly angered. And then we've got the best friend of the killer T cells, which are the suppressor T cells. These are the friends that are like the best friends of the killer T cells. They're like, no, you need to stop. Okay, you've done it, you need to stop. And they're the ones that are actually going to repress the immune response. They're going to repress and control the killer T cells to not actually go ahead and kill all the cells in your body. Because the killer T cells are going to go ahead and want to kill off those pathogens. The suppressor T cells go, no, we need to stop the immune response once you're done. And that's kind of the, I guess I like to call it like the four friends of the group. We've got a helper T cell, the one that's always there, kind, really fast to react. Memory T cell, the organized type A, that's like, we need to remember this. Then we've got the killer T cell and the suppressor T cell, which are like, the two best friends in the group that are like, one's like really aggressive, really angry, really quick to react, and then the suppressor T cells like, no, you need to calm down. So that's the T cells. Now the B cells, we've only got two of those. We've got the plasma B cells. Plasma B cells are the one that are actually going to start to create the antibodies. They're going to so the B cells differentiate into memory B cells and plasma B cells. Plasma B cells are the ones that actually secrete antibodies, and memory B cells are the ones that also remember the antibody. So we've got, this is called the antibody mediated immunity, and with the helper T cell, it's called a cell mediated immunity. So hopefully that makes sense. Again, please ask away any questions. So essentially what is happening here is that the first response, the primary response when you're first infected, is going to be slow. It's going to take time. You can see here, days after antigen exposure, it is going to be at its highest. It's going to peak at like a week after. But however, as soon as you get the a repeat infection, we see that immediately the response is faster and it's more potent. So 3 days into the 3 days after the antigen exposure, we are already peaking. Like, we can already see the plasma cells doing their thing, secreting the antibodies, and then we've got long-lived plasma cells in the bone marrow and memory B cells. So they sustain, and we now have even more memory B cells that are actually going to remember what the antibody was after the second exposure. Okay, so vaccinations are effective because in the primary response, the B cells try to find the antibody, right? This is where, we actually, with vaccines, with the general vaccines, we're putting a part of the virus into the person's body. With mRNA vaccines, they're different. So COVID-19 vaccines are the mRNA vaccines. But essentially what we're doing is, with the primary response, we're trying to elicit that primary response before the infection even happens, because we're putting a small amount in. We want to see the primary response so that when you actually get the infection, your secondary response is going to be fast. It's going to be really, really effective. Okay, so this is a summary for us. This just outlines all the differences between each one, and I'm just going to move on just so that we can actually spend time on module 8. So key things to remember: cells and their roles. Remember B cells versus T cells, helper versus killer cells, so on. The analogy of the friend group just always helps me remember it. If that helps you, go for it. If you find another way to remember it, go for it. What are antigens, antibodies, key molecules? So the MHC2 receptors, for example, these are the ones that are on the macrophages, and these are the ones that are going to display the antigen. Cytokines, and these are the ones that are responsible for actually killing off the responsible in calling and then killing off the pathogens, and receptors, and remember the flow of events. It's logical, try and build it up from the basics. So let's move on to prevention, treatment, and control. So this is where we are looking at how are we actually limiting the spread of disease. So with local, we're trying to do small things day to day. Herd immunity, obviously this is something that has come up not now, but 2 years ago. Yeah, 2 years ago it was in the mainstream media a lot when certain countries tried to achieve herd immunity against COVID-19. We saw that it wasn't the most effective way to do it, but obviously in local communities over time we can see this happen. Personal hygiene, healthy practices, and also spreading public health information. Then on a regional level, we're trying to monitor environmental conditions. We're trying to improve identification as well through surveillance. For example, with COVID-19, it was knowing every day which areas were where potential cases happened, and then creating appropriate responses. Finally, on a global level, we're looking at quarantine, communication. For example, the World Health Organization was a key player in the communication during COVID-19 pandemic. And we're also looking at monitoring affected and potentially affected individuals via their travel cards and so on. Obviously, travel was limited with COVID-19, but this happened with Ebola, where during Ebola in the Ebola pandemic in it was an epidemic actually. During, I think it was 2014 or 2013, 2014 I believe. If you were every international every international passenger had to obviously fill out a declaration card saying that they actually hadn't traveled to Africa in the past week or so. I can't remember. I know I traveled, I was overseas at the time, so when I came back, we had to do the whole thing at immigration. So that's why I actually remember that one thing. But that was they did that in order to actually track any movements. So, what is the incidence and prevalence of an infectious disease? What does that mean? Incidence refers to the number of new cases over a specific period, like in a week, in a month. Prevalence is the overall number of cases, or the proportion of population that is affected by disease. So, for example, with COVID-19, the prevalence is going to be, I can't remember the number of cases, but I think, it's going to be the number of cases since the beginning of the pandemic. Incidence could be the number of cases in May, for example, in June, and so on. And then mortality refers to the number of deaths within a particular population as a result of the disease. So, last stop point of the module is asking us to look at the contemporary application of Aboriginal protocols in the development of particular medicines and biological materials. So, smoke bush, which is endemic to southwest Western Australia, is a traditionally used medicine by the indigenous peoples of the region. In the 1960s, the US National Cancer Institute was looking for cancer-fighting molecules, and the Western Australia government grants them a license to actually collect the samples of the plant. And in 1980s, during the AIDS epidemic, smoke bush was screened, and they found that it contained some molecules that helped combat the HIV virus. And the US Department of Health and Human Rights filed a patent for its exclusive rights. The WA government was paid 1.65 million at the time, and in the process, I think it's still ongoing, but if it was successful, they could be paid up to $100 million in royalties per year. Now, obviously, the whole concept came from Aboriginal knowledge and their knowledge system, and the indigenous peoples, they were not consulted, they did not get any royalties or compensation, and there was no acknowledgement given to them, using the plant already for so long. So that is module 8. Now that is module 7. Let us overview module 8. Now if you have any questions, please ask away. Okay, awesome. So, what is homeostasis? That is the big question of Module 8. Now, the inquiry question is looking at how is an organism's internal environment maintained in response to a changing external environment. So let's have a look what's happening here. Now, this module, there's a lot going on here. Firstly, the first point that we're looking at is homeostasis. We're looking at how is an organism's internal environment maintained in response to a changing external environment. Causes and effect. This is where we look at do non-infectious diseases cause more deaths than infectious diseases. Epidemiology, we're looking at what epidemiological studies are and what they involve. Then we look at prevention, so how can non-infectious diseases be prevented? And finally, we are looking at technologies and disorders. How can technologies be used to assist people who experience disorders? So, homeostasis. Think back to year 11. Always love doing that, don't we? Thinking back to year 11. Homeostasis refers to the steady state of internal physical and chemical conditions which are maintained by living system. So we need to have consistent conditions within the body for everything to function properly. The body is amazing in how many different things it's doing and how many different systems are doing amazing stuff and working together to sustain us. And we need homeostasis for things like enzymes, for enzymes to work. Most things in the body are working thanks to enzymes. Obviously, to maintain cell conditions and cell metabolites, we need the body to be in a stable internal condition. So essentially, this is where we need to regulate things like temperature, water levels, waste levels, and the levels of nutrients. So what do we do as a result of that? The body does, or rather uses, uses I think is a better word, something called a negative feedback loop. So what is that? A negative feedback loop is essentially when we are trying to counteract the change to the body in order to return to the normal conditions. So homeostasis is about maintaining a stable internal environment regardless of the changing external conditions, right? So even if you're out in the cold, it's really cold on a windy day, you still want your body to maintain its stable 37-degree temperature. If it drops below that, we start to see things like shivering, for example, and that's happening because the body is trying to return back to the normal state. So feedback systems are designed to counteract the changes and to return to the normal condition, and they oppose a stimulus. A negative feedback loop is opposing a stimulus, right? If the temperature has gone up, right, we want to bring it back down to the normal temperature. If it has gone down, we need to bring it back up to the normal temperature. So it's kind of opposing what has happened. So let's have a look at the temperature negative feedback loop. So essentially, we start off here with knowing that the normal body temperature is around 37 degrees. However, we notice that there is an increase in temperature. So the stimulus is that the temperature increases. Immediately, receptors, so actually before I do that, sorry, I'll just quickly walk you through this diagram here before we go on to the next one. So what happens is we've got a stimulus, which is the change that is happening. Something has happened, that's a stimuli. Then we've got sensors in the body. Now, sensors, they could be, for example, those receptors that we just looked at a minute ago, or second ago rather. And then we've got a control center. And the control center is going to then dictate how we react to it and send the signals to the effector. And as a result of that, we're then going to see the change being counteracted. So this is what is happening over here. We've got a normal body temperature around 37 degrees Celsius. Now, there is an increase in temperature that is immediately going to the sensors. In this case are the receptors, right? The thermoreceptors in the skin, they are the sensors. They're going to then send message to the control center. The hypothalamus is a control center here. The hypothalamus is located in the brain, and messages are sent to the hypothalamus via the nervous system. Then effectors are going to be they're going to receive the signals from the control center and the signals are going to be like, hey, you need to start sweating, or we need vasodilation to happen because the temperature has gone up. We need to lose the heat and return back to 37 degrees Celsius. And we're going to start to sweat. We will see vasodilation, and then we'll also see increased metabolism. So increased metabolism is going to burn off the heat, and that is going to cause a decrease in the temperature. However, if there is a decrease in the temperature that is also going to be detected by the thermoreceptors in the skin, then the control center is going to be notified. The control center is then also going to notify the effectors and tell them that, hey, you need to start shivering, there has to be vasoconstriction, and also goosebumps as well. And that is then going to cause the body temperature to increase and return to 37 degrees. And that's the whole cycle. Think of it like an 8, right? So we start off, the first half here is if the temperature increases, second half here is if the temperature decreases. Now obviously the structure of it, like, if one happens at the others, like, it doesn't have to be that way. Like, what I mean by that is that the decrease could be up at the top here and the increase could be at the bottom, but the point here is that we see the way in which it's a loop. One thing, we go from receptors to the control to the effectors, and all of these different body parts work together in order to then bring your internal environmental condition, to bring your internal body condition back to normal, and that is really important. So key elements here are the sensors, the receptors, they're the thermoreceptors. We've got a control center, and that is the hypothalamus in the brain. And then we've got our effectors. So our effectors include sweat glands. We see sweating. Skeletal muscles are also the effectors, because the skeletal muscles will then start shivering. Smooth muscles will either vasoconstrict or dilate. They're going to cause, vasoconstriction or vasodilation. And muscle cells, they're going to increase or decrease the metabolic rates. Okay, that's the temperature homeostasis. Now the syllabus also wants you to look at glucose homeostasis. So glucose homeostasis, I think with temperature it's probably I won't say easier, but because we kind of know what is happening, and we've looked, you know that if the temperature goes up, you're going to start sweating, or if the temperature decreases and it's really cold, you're going to start shivering. So it may be a bit more intuitive than the glucose homeostasis, but I kind of want you to follow along, maybe note down these keywords, and I'll try to take you through all of this really slowly. So, homeostasis. Our normal blood glucose levels are about 90 milligram per 100 mils. That I always find that a bit hard to remember, so you can remember it as around 4 to 5 millimolar. That is what our homeostasis levels have to be, about 4 to 5 millimolar. However, there can obviously be imbalances when you eat a meal or you don't eat a meal for too long. Now, if you eat a meal or you eat something really sugary, let's say, so that's going to be a stimulus, and our stimulus is going to be the rising blood glucose levels. You've had a big meal, high carbohydrates, so there is rising blood glucose levels as a result of that. So what happens is that high blood glucose levels, they are detected by the insulin secreting cells of the pancreas. So there are beta cells in the pancreas that secrete insulin, and these beta cells are going to immediately recognize that the blood sugar is increasing. Now, what insulin does is that insulin is going to decrease the blood levels. It's going to decrease the blood glucose levels. We don't want those high blood glucose levels. We don't want the blood to be so concentrated, because that is going to affect the rest of the bodily function. It's going to mess up with metabolism. It's going to mess up with other processes. So we need it to be between 4 to 5 millimolar. And so what happens is, the beta cells in the pancreas, they are going to see that the blood glucose levels are high, and they're going to secrete insulin. What insulin does is that once insulin is released into the blood, it is going to have the muscle cells and the liver take up glucose. So most body takes up more glucose. We don't want that much blood glucose, so we want the muscle cells and so on to actually, muscle and adipose cells, so those are our fat cells, we want them to take up the glucose. So they're going to start taking up the glucose. Along with that, the liver will also take up glucose. But what the liver does is that it won't store glucose as is. It is going to change the glucose into something called glycogen, and glycogen is basically like a big chain of glucose, like chains of glucose. Like you've got 12 to 14 molecules of glucose around in one glycogen molecule, or in one glycogen unit, you've got like 10-12 glucoses attached. You don't need to know that, but that's just to explain to you why the liver is doing that. It's taking up glucose and storing it up and chaining it up as glycogen. And then as a result of that, the blood glucose level will decrease, and it will decline to a set point, and then the stimulus for insulin release will diminish, because we don't actually want high levels of insulin in the body at all times, because the glucose levels should not drop below 4 millimolar. And as a result, once the other body parts take up glucose, insulin release will stop, and the blood glucose levels will return to normal. That is what we want. They're going to get to that point where we need them. Now, let's say you haven't had a meal in a long time, okay? You haven't had a meal for the past or you haven't eaten anything for the past 4 hours, 3 to 4 hours. There's going to be a decline in your blood glucose levels. And what that means is that the low blood glucose level will be detected by the glucagon releasing cells of the pancreas. So the pancreas and the glucagon releasing cells are your alpha cells. So the two hormones that you need to remember here firstly is insulin and glucagon. Insulin is the one that is released by the beta cells of the pancreas. So let me say that again. Insulin is a hormone which is released by the beta cells of the pancreas, and it is released in order to control and decrease the blood glucose level. So when glucose levels are high, insulin is released. Glucagon is a hormone that is released when blood glucose levels are low. You could think glucose levels low equals glucagon. Yeah, maybe if that will help. I know it's not really a mnemonic or anything, but yeah, I don't know. Try that. Maybe try and make something up for it. But essentially, we've got low blood glucose levels, glucagon is released, and glucagon is released from the alpha cells of the pancreas. So beta cells release insulin, and alpha cells release glucagon. And then from there, once the glucagon releasing cells of the pancreas are stimulated to release glucagon into the blood, it is going to target the liver. It's not going to target the muscles or adipose tissues just yet. It will only do that when you're like in starving conditions, or when you're like, for example, like exercising at a really high intensity. But that's a whole different topic. That's not related to this. That's like metabolism and stuff. What is happening is that essentially glucagon is mainly going to target the liver. It is going to ask the liver to actually break down those glycogen stores. So remember, we stored glucose as glycogen, and we want to now break down the glycogen stores to then increase the blood glucose levels. And once the blood glucose levels start to increase, the stimulus for glucagon release will go down, and as a result, we're going to come back to the normal body glucose level, and that is the whole loop. So the key elements here are, firstly, we've got pancreas. They are very important organs because they are detecting glucose levels, and they are secreting both insulin and glucagon. So pancreas secrete both, but obviously different cells do that. Beta cells are going to secrete insulin. Alpha cells are going to secrete glucagon. Now, the liver It stores glucose as glycogen. That is what the liver does. And other tissues are greedy idiots that just like to use up the glucose. So liver is really important because because it is storing glucose as glycogen. Glycogen is basically like there's lots of glucose chains attached to a glycogen molecule. Now, the two hormones that we talked about, glucagon is released when we have low blood glucose, and it essentially goes to the liver to say, hey, could we get some glycogen happening because the body needs it. And like I said earlier, the body, if the blood glucose levels go below 4 millimolar, that is essentially, yeah, really bad. Really, really bad. So it has to stay in that 4 to 5 millimolar level, because remember, your brain is using a lot of glucose. I think it uses like 120 milligrams per day. So it's using a lot of glucose. And insulin is released when the blood glucose levels are high. We don't want that. We don't want the blood to be so concentrated because it's then going to mess up with other processes. So we go to the liver and say, hey, do you mind, keeping some of this glucose with you, like that kind of thing. So that's the glucose homeostasis. They can ask you, for example, in papers, one of the questions in the sample paper that NASA, sample additional questions that NASA released, was what is happening in a glucose chain. Actually, I think it was like a graph. You had to show you had to plot glucose levels and then explain what is happening. And then the second part of the question was, how is glucose homeostasis different to temperature homeostasis? Okay, so the second dot point of this first inquiry question is asking us to look at the various mechanisms by which organisms maintain their internal environment within tolerance limits. So this is where you would talk about trends and patterns in behavioral, structural, and physiological adaptations in endotherms that assist in maintaining homeostasis. So you want to have examples of behavioral, structural, and physiological adaptations. A lot of that is your year 11 stuff. So you can easily definitely borrow from your notes there. The second part here, we want to look at the internal coordination systems that allow homeostasis to be maintained. This is where your central nervous system comes into place. The nervous system constituting of your central and peripheral nervous system. This is where that comes into play because through that the homeostasis is being maintained, hormones are being produced. There's a lot going on in the brain that is controlling the whole thing. And then finally, mechanisms in plants that allow water balance to be maintained. This is where we're looking at the transpiration, cohesion, adhesion theory. So definitely check that out as well. Revise that. You've gone through it in year 11. You should have gone through it in year 11. But if you have any questions regarding them, please feel free to ask. So let's have a quick look at this hormones specifically. So this is part of the second dot point. So the internal coordination systems that allow homeostasis to be maintained, including hormones and neural pathways. So hormones are signaling molecules. They regulate physiology and behavior, and they are produced by the endocrine system in the glands. So some glands that do that include the adrenal cortex, thyroid, testes, ovaries, mammary glands, bones, mammary glands, and uterine muscles, and also kidney tubules. So the hypothalamus plays a big role in it, and the hypothalamus is connected to the pituitary. We've got the anterior pituitary and the posterior pituitary. And essentially the anterior pituitary produces its own hormones, while the posterior pituitary produces and the posterior pituitary secretes the hormones that are produced by the hypothalamus. But you don't need to know that in any detail. You just need to know that they play a huge role in the regulation of hormones. The hypothalamus At the pituitary, they secrete and release hormones. I think it's good to have some examples, like to know, for example, ADH, oxytocin is another one and oxytocin is something you would have looked at in module 5 as well when you were looking at fertilization and mammalian birth and so on, or rather birth in humans. What else? The GnH, FSH, thyroid stimulating hormone, so that essentially then goes on and activates the thyroid releasing hormone, and obviously that releases thyroid. And the adrenal cortex releases the adrenal glands release cortisol, which also plays a huge role in the fight or flight response of the body. But that's just some extra information if you're interested in knowing the context of the diagram and some of these. So hormones are signaling molecules. They are transported around the body using circulatory system and lymph system. And examples include insulin. It signals the cells to uptake glucose, just like we looked at then in the glucose homeostasis. Glucagon releases, stimulates the liver to release glucose. Leptin decreases appetite. Ghrelin stimulates appetite, and aldosterone is responsible for the reabsorption of sodium and also water to some extent, but usually sodium in the kidneys. Okay, so how is the whole thing happening? Essentially, we go through a process called the signal transduction. And what happens is we start off here. So this is the plasma membrane. So this is the inside of the cell. This is the outside. We've got extracellular fluid on the outside. This is a hormone. It's going to come and it's going to attach to the receptor on the plasma membrane. From there, it is going to then cause transductional changes. So we're going to see transductional changes. So I had to click. Sorry, I was just going through the whole diagram. Okay, so you see reception. That's the first step when the hormone is going to bind to the receptors on the cell. Transduction is the second step where we see a signaling cascade in the cytoplasm. So basically, kind of like a domino, right? So dominoes rather. One thing causes another, so the signaling transduction, and then finally, step 3 is when this transductional then causes changes inside the nucleus. It triggers a response, and that response is generally a gene being switched on or off. And from module 5, we know what that means. That means, for example, if a gene is switched on or off, that is then going to affect protein synthesis and transcriptional and translational factors as well. Again, feel free to ask any questions. So generally, in summary, hormones, they work by regulating expression of certain cell components and hormones. They allow the body to coordinate changes to cell structure and function. They can lead to larger changes in your tissues and organs, and hormones can control processes like metabolism, digestion, respiration, sleep, growth, development, reproduction, and mood as well. That's the first dot point done. The first inquiry question done. Let's have a look at the second inquiry question, where we have to look at the causes and effects. So, do non-infectious diseases cause more deaths than infectious diseases? So, we are going to, the syllabus wants us to look at look at genetic diseases. Now look at the wording here, including but not limited to. So we definitely want to look at genetic diseases, but you may choose to do some, for example, in class, some other examples as well. So a genetic disease, as evident through the name, is a disease that is caused by an alteration to a gene or a set of genes. So a single gene disorder is when there is alteration to one specific gene. For example, in cystic fibrosis, Tay-Shay, sickle cell anemia, that is what happens. There's an alteration to one specific gene. Chromosomal abnormality is when there's and again, as the name suggests, is when there is an alteration to the chromosome number or its structure. Examples include Down syndrome. Then mitochondrial disorder is when, again, the name, as evident in the name, is when there is an alteration to the mitochondrial DNA, like LHON, so Leber's hereditary optic neuropathy disease. Finally, multifactorial disorders. This is where we see a combination of genetic and also environmental factors contributing to a disease. So heart disease, Alzheimer's, cancers, all of them, they obviously have genetic susceptibility, but they also can be heavily influenced by your environmental factors. So causes of genetic disease, of course, mutations. This bit is linking very strongly back to module 6. If you have a good understanding, and so this is where, it's like a cross-module thing. And one thing that you'll notice in the HSC papers is that your higher order questions, your 8, 9, 10, 11, 12 markers, they are going to be cross-module to some extent. Some of them can be, they don't necessarily have to be, but so far we've seen that a lot of cross-module questions have come where they are asking you to consolidate your knowledge from, let's say, module 6 and 8 or 5 and 6. So, looking at the causes of genetic diseases, we've got mutations, we've got, and they include random mutations, error in the gamete formation, and of course, exposure to different mutagens. Inheritance from parents can also cause genetic diseases, and so can acquired changes. So, in your study notes, have a definition, a brief explanation of each. What is a single gene disorder? What is chromosomal abnormality? And 2 examples of genetic diseases that are caused by the different factors, and include specific details about the individual causes and effects of the disease. Okay, so let's have a look. Point mutations. Point mutations are the ones which change or affect only one or very few nucleotides within the genome. And chromosomal mutations, on the other hand, can affect entire chromosomes. They can affect certain parts of the chromosomes, and generally, like a significant aspect of the chromosome. So we can see things like deletion, where we're deleting a certain part of the chromosome, duplication, inversion, and translocation. So it's like switched over incorrectly. So, and remember, with this, you want to know the different types of point mutations and their effect, like missense, nonsense, silent, and so on. And know examples for your chromosomal mutations as well. Okay, so the causes and effects. So with point mutations, there are 2 types. There's either substitution, where we are substituting when there's substitution of a certain part of the codon. So here we are replacing the G with the U, and that's going to change the amino acid produced. A frame shift mutation is when there's a downstream shift in actually the reading of the codon. So this is where, for example, here I'm adding in a C between the A and the G, and that is then going to shift the sequence down, and so I'm going to get a completely different set of codons, as you can see. Deletion is when we are deleting a certain codon, and that is then going to cause, again, a differing amino acid. And the effects are missense, nonsense, and silent. So missense is when there's that, as we can see here, from the substitution, there is going to be obviously editing to that codon, and it will cause a different codon to be added to the chain. Nonsense is when we are introducing a premature stop codon, and silent is when there is no effect of the mutation on the genome, because here we replace the C with a U. However, it's still going to lead to the same amino acid, so it's not actually causing any issues to the genome. So causes external mutagens, DNA replication errors, and also when we see issues with the repair system, the DNA repair system not working. Effects include, effects can be silent, missense, nonsense. We see creations of SNP, so single nucleotide polymorphisms. We see the creation of new alleles, and it can be potentially beneficial, potentially negative. Just because a point mutation is small, just because it's like the shift of one codon does not necessarily mean that it's not going to have an impact, right? It can have a huge impact, and an example of that is cystic fibrosis. So those are genetic diseases. Now let's have a look at diseases that are caused by environmental exposure. So environmental diseases, as the name suggests, are caused as a direct result of an individual's environment. And they include exposure to either be, and that can include exposure to chemical or physical substances. So lifestyle diseases are those caused by lifestyle behaviors and choices. They include cardiovascular disease, alcoholism, smoke-related diseases. Diseases that are caused by physical factors include exposure to radiation, for example, and diseases caused by exposure to chemicals, whether that may be through work or, like in general environment, and they can include exposure to toxic metals as well. So some diseases can happen as a result of overexposure, for example, skin cancer or heavy metal poisoning. Or they can also happen due to underexposure, like deficiency in adequate exercise, and then also vitamin D deficiency. So if you're not in the sun enough, if you, like, the biggest contributors to vitamin D is sunshine, but then again, if you overexpose yourself and you're not safe in the sun, then that can overexposure over a long period of time can then cause skin cancer. So this is again where we need to have that balance, and that balance comes in. So examples of such diseases include Minamata, which is the ingestion of large amounts of mercury, and there's a link there in the slide so you can use to get some more information, and mesothelioma, so that is cancer as a result of asbestos exposure as well. So let's have a look at the causes and the effects of genetic diseases. So they can happen, firstly, so let's have a look at structural chromosome and mutation. So we see deletion, so part of the chromosome has been deleted. Inversion, that's been inverted. Upside down, translocation, we see that switch. And finally, duplication, so that part is added. So examples include Jacobson disease syndrome, hemophilia A, leukemia, and also the Charcot-Marie-Tooth disease. So usually they are a result of errors in meiosis. For example, if crossing over occurs incorrectly and chunks of DNA screw up, that can cause structural mutations. And if sister chromatids are separated incorrectly during anaphase, that can also cause a number of mutations. So remember how we see the spindle fibers that are attached to the chromosomes, to the chromatids rather, and when they have to be separated, if they're not separated correctly, we can see different diseases. So sometimes there can be an extra number of chromosomes in, for example, the in the sex cells in chromosome number 23. So effects are usually severe, and that is because chromosomal mutations involve a large number of genes and changes to those genes, and that's going to then cause changes to the protein sequences and so on. So mutations in the chromosome numbers, there is aneuploidy, which is when the overall chromosome number of the offspring is different to the parent chromosome number. So in Down syndrome, there are 47 chromosomes instead of 46, and that is because chromosome 21 has 3 copies, and this is what I was just mentioning in terms of the sister chromatids not separating correctly, and that can cause an uneven number of chromosomes as well. So that's aneuploidy. Now, polyploidy is when an organism contains more than 2 sets of homologous chromosomes. So a fetus, for example, could have 3 copies of every homologous chromosome instead of just 2. Okay, so nutritional diseases. Nutritional diseases can be caused by excessive nutrient consumption, like, for example, cardiovascular disease, type 2 diabetes, stroke, excessive consumption of carbs, fatty acids, and so on, or rather fats, and then that then leads to fatty acids, inefficient nutrient consumption, so malnutrition, scurvy, which is the lack of vitamin C, and anemia, which is the lack of iron. And then finally, problems with digestion also comes under nutritional diseases. So every country in the world is affected by malnutrition. So it's quite a big issue in most parts of the world. And countries with a burden of at least one of for example, child stunting, anemia in adult women, and overweight in adult women can be caused by malnutrition. So, single burden, for example, in Russia, Australia, America, at least double burden, parts of Africa, South America, and a triple burden in parts of Africa. So we can see what a big disease it is and what a significant effect it's having around the world. Okay, now cancer is also another one that you'll be looking at, and again it's mentioned in the syllabus. So cancer is a disease which occurs due to the abnormal cell growth in an which occurs when abnormal cells grow in an uncontrolled manner, and this is where we see changes to fundamental molecular functioning of a cell. So it's uncontrolled and there is infinite cell division. Now, mutation to normal cells can also happen as a result, and inability to regulate cell growth. So basically, cancer is that when we see, and we will talk about benign and malignant tumors, but essentially when we see the cancer spread throughout the body, and it can then grow. It can grow really rapidly and spread around the body, and it can grow really rapidly, the cells divide really quickly, and alterations leading to cancer phenotype include indefinite replication, evasion of growth suppressors, so these cells can then actually also resist cell death. So like we spoke about in module 7, apoptosis, when there's cell death, these cells can then become powerful enough to actually resist cell death. There is induced blood flow. They can also induce blood flow to the tumor site, that's called angiogenesis, and also invade other tissues, and that is metastasis. So as you can see here, we've got cancer in one part of the body, and it's slowly starting to spread to other parts of the body. And the effect of this uncontrolled proliferation is that there's a reduction in system normal organ function, there obviously invasion of tissues, and also there's growth to clog or impede other bodily systems. So mutagens, we've spoken about them. So we spoke about, and again in module 6 you would have looked at them. So electromagnetic radiation, these are highly penetrative waves which interact with and ionize atoms, and essentially their high energy alters the DNA bonding, and they can cause the rearrangement of the structure. So a big example of that is UV light. The radiation from the sun can be harmful in high doses, and it can cause pyrimidine dimers when, for example, the bases, instead of sort of being stacked, how the bases are sort of stacked against each other like this, you've got them down here, just imagine my fingers sort of being the bases here. So pyrimidine dimers is essentially when they turn their shape and adjacent bases connect with each other. And I think I have an image there, not sure. Okay, I don't have an image there. I think I did. But anyways, definitely search that up to get a better, view of it. But it's essentially when adjacent bases sort of they change their shape and connect to each other. Chemical mutagens, they include radioactive agents like uranium, and they release radiation to cause structural disruptions. Metals like arsenic and nickel, they can affect DNA replication, and also they can affect the DNA repair system. Now, our repair system is really, really good at fixing any DNA, like any mistakes in the replication process. It's really, really good. A lot of mutations happen as a result. So if we see that there is an issue with the repair system that is going to cause mutations, and metals can affect the repair system. Intercalating agents, these are, for example, ethidium bromide, they can cause frameshift mutations. Then naturally occurring mutagens include viruses. They are going to change our cell machinery by replicating their own material within our cells. Bacterial infections, they can cause inflammation. They can inflammation can then they can cause inflammation, which reduce repair and increase mutation. So remember, as we spoke about, when there's a bacterial infection, inflammation happens, blood rushes to the area, and that can cause reduced repair and increase mutations as well. And finally, transposons, they can cause chromosomal mutations. And here's a summary for us: what the causes are of each and what are the effects. And I've got some examples here for you. Definitely remember the examples as well. Okay, I think we're going really well according to time. So let's have a look at epidemiology and why are epidemiological studies used? Why do we care? So epidemiology is the branch of medicine that is looking at the incidence, distribution, and possible control of diseases and other factors that relate to health. And the study of diseases that affect a large proportion of the population. Those are generally what we study, because obviously, like if a lot of people are affected by it, we want to study it, find a solution. And it describes the pattern and causes. That's really important, pattern and causes. So it's not just about finding it and fixing it, but it's also about seeing where are the patterns, how is it happening, what can we actually do to prevent these diseases. So studies tend to be observational rather than experimental, and that is because an observational approach will collect information about the extraneous factors, but it doesn't exclude them. Therefore, conclusions sometimes can be false with observational studies. Usually, we can only go to prove that an agent could have caused the disease, or could have caused an effect in any case, not that it actually did. And that's the difference between correlation and causation. Now, with an observational approach, we can say, okay, so if you're seeing for example, in people who smoke, we're seeing in patients who have lung cancer that they had a big history of smoking. We can say that potentially tobacco smoking is going to contribute to lung cancer. And obviously studies over time has really, really strongly indicated that, but we can't always say, like in observational studies, like directly, that it actually causes lung cancer, because some people who don't smoke, who've never spoken in their entire lives, they can still have lung cancer. So there's that difference between correlation and causation, so we've got to be careful with that. So analyzing patterns. The patterns that we sort of tend to analyze include, for example, what is the difference in incidence between countries? Are some countries more, like, do they have a higher prevalence of a disease in comparison to others? What is the difference between incidence? What is the difference in incidence between genders? What is the difference in incidence between occupation? For example, workers, like for example tradespeople who are working out in the environment, out in the open all day long, for example construction workers, they would obviously have a higher rate and higher exposure to sun, which can lead to high susceptibility to things like skin cancer. So in comparison to someone who works like in a corporate job where they're inside an office all day. So there's also, again, difference in incidence between occupations. What is the difference in incidence between lifestyles, and what is the difference in incidence between age groups? Are more age groups susceptible to it than others? So we are essentially then looking at what is also the reason for these observed trends. These are the trends, but why are they there? And the example I gave you earlier with countries, with occupations, it's because of certain environmental and lifestyle factors. So let's have a look at type 2 diabetes. This is the example we're going to study because the syllabus wants us to analyze patterns of non-infectious diseases in populations, including their incidence and prevalence, including but not limited to nutritional disease. Now, type 2 diabetes, and again, you can definitely reuse this example, and it pops up, I think, twice or thrice in the module, in module 8. And type 2 diabetes is a condition in which the body becomes resistant to insulin, and gradually it is unable to produce insulin, and that is going to cause a buildup of glucose, and that is going to cause damage to systems in the body. So think back to the glucose homeostasis that we looked at. Now, insulin, remember, is the hormone that is going to control glucose levels. It is going to control glucose levels. It is going to reduce blood glucose levels by sending glucose off to the muscles and liver. However, if you, let's say, have, there are two types of diabetes, of course. Type 1 diabetes is when your body's natural ability, like, that is essentially, type 1 diabetes is like an autoimmune disease where your immune system attacks the beta cells of the pancreas, and the beta cells produce insulin, so your body doesn't actually produce the insulin that it needs in order to control the blood glucose levels. That's why we put in exogenous insulin, and that is something you would have looked at in Mod C when you looked at how you grow insulin from circular bacteria. So type 2 diabetes is now when, if you've got a very unhealthy diet, a sedentary lifestyle, and also genetic factors, that is going to over time lead to type 2 diabetes. So if you're constantly eating unhealthy, high foods, with high amounts of sugar and so on, it's going to start off by, firstly, your body becoming intolerant to insulin. So, like, for example, in the early stages, you're going to need more insulin to actually reduce the glucose levels. And over time, if you keep that up, if your body keeps requiring lots and lots of, lots and lots of insulin to control the glucose because you're constantly eating unhealthily, that is then going to cause, over time, your body to become resistant to insulin, and ultimately that is going to cause your body to actually stop making insulin. So it is a big disease. It is quite significant around the world. In US, there are about 1.4 million new cases every year. Incidence of diabetes has quadrupled over the past 3 decades, and there are predicted increases in the cases between 2010 and 2030. Now, 20% of those cases will be, as we predict, will be concentrated in developed countries, while 69% will be in developing countries. And just by looking at that difference between developed and developing countries, we can see how, for example, in developed countries, we'd have, for example, you would have more treatments available, you'll have more resources available, and also perhaps more preventative programs to stop it in comparison to developing countries. And also there's differing lifestyle and like food habits as well that are going to contribute to that. Now, the prevalence, 1 in 11 adults between the age of 20 and 79 had diabetes in 2015, and 90% of those were type 2 diabetes. So again, type 1 is autoimmune. It is happening because your body cannot actually produce the insulin because your immune cells are attacking those beta cells and stopping them from producing insulin. However, type 2 is when over time your body has just grown resistant to the insulin because you kept eating unhealthy over time, and it started off by your body needing more insulin to come back to the normal glucose levels, and over time the body just kind of goes, yeah, okay, kind of give up now, and then it becomes resistant to insulin. So we are expecting that the prevalence will rise to 642 million by 2040, and there is an epicenter of this epidemic, and that's in Asia. So China and India are the top 2 epicenters of diabetes, and that is contributed to the diet, to the high carb diet most of the time. Now, mortality, 5 million deaths per year, 1 death every 6 seconds. So, sorry, I was gonna sneeze. Okay, mostly due to diabetic complications such as cardiovascular disease. So, cardiovascular disease, so your heart disease and so on, they're quite closely linked with diabetes. And yeah, and type 2 diabetes is expected to become the 7th most prevalent cause of death by 2030. Sorry, I need to sneeze, but it's just because I'm talking, I'm sure it's just not coming. But anyways, so here is a map to show you the number. So in essentially the pink is where we've got the most number of cases. So US, parts of the US, we see India, China, so large numbers. 69.2 million, 109.6 million, 29.3 million, and then we are seeing rising cases in South America, in Europe over here with Russia, and then 1 to 10 million in America, parts of America again, Australia, parts of Africa, and South America, and then pretty small and controlled numbers in certain parts of Africa and also certain parts of Europe as well. Okay, moving on then. Here's some data for us. So type 2 diabetes, Australian data. So this is where we're now looking at the differences in populations. Maybe I'll pull up my pen or something. I don't actually have a stylus to write with, so I'm going to use my... Oh, it's not going to let me... Oh, no, it is. Okay, amazing. So, let me come over here. So, prevalence of diabetes among persons aged 18 and over by age and sex between 2014 and 2015. So, we can see that men generally have a higher number of cases, and we see that the number of cases increase once you go over 50. So, 50 plus, the numbers start to increase, and the numbers are higher in men. Prevalence of diabetes in terms of remoteness and socioeconomic groups. So those living in major cities, in a regional and outer regional. So we can see that outer regional has a higher number. And then in terms of socioeconomic status, socioeconomic groups, the lowest, so group 1, which is so we go from lowest SES to the highest. So group 1 has the highest prevalence, and the lowest prevalence is in group 5, which has the highest SES. And again, think about why that is. Lifestyle factors, right? So low SES, we may not be able to get as much healthy foods, organic fruits and so on. So you may be relying on junk food occasionally, in comparison to group 5, where we may be able to, we may have access to like gym memberships and stuff, do more exercise, have a lifestyle that allows us to be more active, and obviously eat healthier as well, because we can afford that. That's a difference between as you can see, between SES, and then again, major cities and inner regional, they're pretty similar. I mean, in terms, I think the big difference here is in the women. So major cities, the number is low in comparison to inner regional. So essentially, the difference here could be between, the difference here could denote to, for example, access to health services, access to information, and maybe different programs as well. And that could be the big difference, and obviously outer regional and remote areas. So this is where, because we're so far out, we may not be able to, or we may not want to go every time, to like get regular health checkups and so on, and especially as you age, right? So that could be one of the contributing factors. Sorry, let me just get rid of my pen. How do I do that? Okay, I think I got it. Okay, so prevention. Now we want to investigate the treatment and management and possible future directions for further research. So prevention refers to the identification of at-risk groups. So things like genetic testing is one way to go about it, and maintaining normal weight, eating healthy, having a varied diet, and exercising regularly, and also things that can be done as prevention for type 2 diabetes, and of course education, in terms of what is actually happening in the body when you're constantly eating high carbs meals that are high in glucose. Treatment and management, so graded response based upon the severity of the disease progression. So if we're starting to see amounts of intolerance and resistance, we can just start off by, lifestyle changes, going on a strict diet, exercising regularly. We also then might start monitoring glucose levels. Medication can also be used. So metformin is a medication which leads to increased glucose sensitivity in tissues, reduced glucose production in liver, and overall decreases blood glucose levels. Insulin injections are also available, and they compensate for pancreatic failure, manually signals to tissues to uptake glucose from the blood. And finally, surgical intervention. So bariatric surgery reduces the size of the stomach. And they kind of progress in terms of like the severity. So you can start off by lifestyle changes, then monitor glucose levels, then take some medicines for it. And then if it's severe, you can resort to insulin injections and surgical intervention. So there are different types of epidemiological studies. So we've got descriptive epidemiological studies. So they investigate causes of a disease-wide patterns of distribution within and across populations. For example, we see if a section of population is affected. We look at the geographical location, the incidence, and the prevalence. Analytical studies, they examine known associations, or they're testing a certain hypothesis. And finally, intervention studies. These are the ones that are used to test the effectiveness or safety of a new treatment or an intervention. For example, a clinical trial of a new drug, or effectiveness of public health campaigns. That would be part of the intervention study as well. So a valid epidemiological study should have a large cohort sample size. So if you get questions that are asking you to, for example, evaluate the study, this is what we're looking out for. We're looking out for a large cohort, large sample size. We are looking at a broad cohort as well, so that we are accounting for different factors like environment, age, gender. Relevant background and medical check of participants is important. Is there a comprehensive questionnaire which is asking participants, their views, how they're feeling, for example, after a certain medicine or something, and peer review prior to publication. So this is a mental checklist, and this is essentially what makes a good study. A good study should have a large cohort. It should have a broad, the cohort should account for a large, large variables. There should be relevant background checks. There should be a comprehensive questionnaire, and it has to be peer reviewed. Okay, let's have a look at prevention. How can non-infectious diseases be prevented? So we are evaluating effectiveness in 5 easy steps. Essentially, we want to be able to implement educational programs and campaigns that are solving their purpose, that are helpful, that are going to have an impact. And with campaigns and programs, I think one thing that is really, really important that you need to keep in mind is that you won't see an immediate effect. It's not like we see a program, implemented last year, and this year you're going to see immediate results. Usually, most programs, they take between 5 to 10, even 15 years to actually show progress, to actually show that over time the number has decreased significantly. So that is something to always keep in mind. But steps that we can look at in terms of evaluating effectiveness include, firstly, we want to see if the methods actually achieve the aim. So do the methods of the program provide targeted and sufficient information? Are the methods actually based on best practice? Or rather, or even if they're based on feedback from previous studies? Is the education information provided clear and understandable? Is the program cost effective? And finally, are there statistics to demonstrate a reduction in the disease incidence? And that is going to be over time. It's not going to be immediate. So let's have a look at this. Slip, slop, slap was, and still is, slip, slop, slap, seek, slide. Also, it's still a very prominent campaign, and it started back in the 1980s. So we had television advertisements and community service announcements, including the well-known catchy jingle, slip, slop, slap. Cartoon Seagull Sid encouraged everyone to slip on a shirt, slap on a sunscreen, and slap on a hat. Message was updated in 2007 to actually seek shade and slide on sunglasses. And then there is also development of sun smart educational resources that are available for both schools and workplaces. And sponsorships of sporting events is another big one as well. So, effectiveness. The rate of melanoma in ages 20 to 24 has fallen from 25 per 100,000 to 14 per 100,000 between 1996 and 2010. Notice this. The program was implemented in 1980, and we sort of saw that decrease, a noticeable decrease between 1996 and 2010. So that clearly tells you that these programs, they take time. They take time for them to be implemented, for the results to show. It's not quick. It has estimated to have prevented more than 43,000 cases of skin cancer in Victoria between 1988 and 2011, and incidence of skin cancer has overall decreased since the implementation of the program. And it's the idea that overall it has decreased, and prevention is always better than cure. So the program has indeed worked. It is regarded as successful. It is believed to play a key role in our shift in attitude towards sun safety, and it's cost effective as well. Now, evaluating effectiveness of current disease prevention methods, and also developing strategies for prevention of a non-infectious disease, including genetic engineering. So this is something you sort of would have looked at in Module 6, and we're now following through with it. So gene therapy, what are we focusing on? So by introducing healthy genetic material, the cell and its offspring can have restored function. We want to restore the function by introducing healthy material. The method is we are identifying the gene that actually causes pathogenesis. We are engineering or designing a healthy allele, and then we are packing that gene into like a viral genome, because remember, the virus is like the viral genome is going to make lots of copies. So we are putting it in a viral genome, we are making lots of copies, we put it into like a gun, which we then and we also coat in organic nanoparticles, and then using that we actually insert the gene into the human. And that can be done in vivo, so it can be done within the living person, or ex vivo, it can be done out of the living, so we can take some cells out as well to try and do it. So you can pick one of these, like cystic fibrosis, hemophilia, and Parkinson's disease, and research that a bit more. So we can correct the CFTR gene in cystic fibrosis that causes cystic fibrosis, or rather mutation in it causes cystic fibrosis. Hemophilia, insertion of healthy gene for factor 8 and 9. So in blood clotting proteins, those blood clotting proteins then lead to like a factor called thrombin and so on, and then thrombin is responsible for clotting. So hemophilia is when the clotting process is affected, so we can change the gene in order to restore the function of these two proteins. CRISPR-Cas9 is something that again we've looked at in Module 6. So there are two parts to it. There's a guide RNA, which includes the nucleotide sequence, which is complementary to the gene we want to actually edit, and Cas9, which is actually going to cut the DNA. It's endonuclease. It's going to cut the DNA in the middle, or rather wherever we need it. It's not going to happen at the side. So the ability to accurately edit genomes give us unprecedented control over, like making changes in living beings. We can edit dysfunctional cells in vivo or in vitro. We can also improve current genetic therapeutics and alter the fundamental genetic cause of the disease to ultimately prevent disease development and stop it from being passed on to the offspring. Embryo screening is the screening of all chromosomes in a developing embryo prior to the implementation before implantation in IVF cycle, and this is where we actually screening the chromosomes and just making sure that the DNA sequencing to identify any defects at a gene level, and it's particularly useful for couples who are known carriers of genetic disorders like Tay-Shay's. So potential future embryo editing, it's not legal in Australia right now, but we are seeing that there's an unprecedented use of these technologies. Okay, this is the last part of the syllabus, so let's go through this, and please feel free to ask any questions. So, how can technologies be used to assist people who experience disorders? You can clearly see now where we are getting that link between Module 6 and Module 8, that we're now just looking at, okay, we know that these disorders have happened. How do we go about improving or rectifying that? So we are going to look at today we're going to look at the eyes and the ears specifically, but the syllabus also mentioned kidneys. So just because of time, just because of time limitations, we'll focus on the eyes and ears today. But if you have any questions regarding the kidneys, feel free to ask in the chat. So we are looking at how these organs function, how these organs are affected, can be affected by a disorder, and what technologies we are actually using to rectify and restore their function. So we've got structure, function, disorder, and technology. So it's that progression. This part of the syllabus really requires you to know, like for example, the structures of the eyes and ears in a lot of detail, because you should be able to link the structure of the eyes and ears to their function. How is it functioning? And then from there, how has the disorder been caused, and how will the technology rectify that? So the important structures here are firstly the outer ear, which includes the pinna and the ear canal, and this is how sound waves are entering the ear. The middle ear refers to the eardrums and the ossicles. So maybe I'll get the pen up again. So, alright, so it includes the eardrums and the ossicles, and essentially waves vibrate the eardrum, and then that vibration causes the eardrum to move against the ossicle bones. And when that happens, when the eardrum is moving against the ossicle bone, the ossicle bone will then beat against the cochlea, and this is the cochlea. It's kind of a little like a snail-like shape, you can remember it that way. And that's the cochlea. So the cochlea, the semicircular canals, and the auditory nerve, they are part of the inner ear. So we've got the inner ear, the middle ear, and this is the outer ear. So we've got the pinna, and then we've got the ear canal. Now, yeah, and then the inner ear is the cochlea and the auditory nerve. So what happens is once those ossicles, these things here, once they beat against the cochlea, which is filled with fluid, like that fluid is moved once the ossicles beat against the cochlea, and those fluid vibrations will cause like bends, and will cause the hair cells in the cochlea to move. Now the hair cells in the cochlea are the ones that actually they actually take the sound waves, and they're lots of hair cells because they I guess interpret the different frequencies of the sound, and then they convert the sound waves into electrochemical impulses. So there's a conversion from kinetic energy to electrochemical impulses, and then the electrical impulses are sent to the brain via the auditory nerve. So the auditory nerve here, as you can see, is connected to the cochlea, and it's going to go straight through. So you should be able to so essentially the ear has two functions: detect the stimulus of sound, maintain balance of the body. And you should be able to talk me through the different structures. What is the outer what is the outer ear, what structures are part of the outer ear, what structures are part of the inner ear, what structures are part of the middle ear. How is sound actually being interpreted? So like I said, we see sound waves energy are directed into the external ear canal by the pinna. They cause the membrane to vibrate with the same frequency. These vibrations are passed onto the ear ossicle. So because they go through to the eardrum beats against the ossicles, the ossicle then beats against the cochlea, and then from there the hair cells in the cochlea are going to convert the kinetic energy of the sound to electrical energy, and then from there those electrical impulses are going to be sent to your brain via auditory nerve, so that you can actually interpret and perceive the sound. So ear disorders, there are two types. There is conductive hearing loss, which is when there is damage to the outer or middle ear, and the effect is that it acts as that ineffective sound transfer to the inner ear. So really important that you remember what part of the ear the hearing loss is in. So if it's the outer ear, that means the sound's just not going all the way through, but it means the inner ear is intact, right? And causes are ear infection can cause it, abnormal middle ear bone growth, so osteosclerosis, and eardrum perforation. Sensorineural hearing loss, on the other hand, is when there is damage to the inner ear, to structures in the inner ear, and that is when there is an ineffective sound transfer from the middle ear to the brain, because things are not moving through the inner ear, so there's that ineffective transfer, so sound can actually not go through to the brain. So causes are so it can be congenital, so loss from birth, and that can be due to genetic factors or diseases during pregnancy. It can be acquired with age, noise, trauma, and also infections like meningitis can also cause it. Hearing loss can be both it can be a mix. So mixed hearing loss refers to conductive and sensorineural hearing loss, a mix of the two. So what are the technologies available? We've got hearing aids. So hearing aids are used for conductive hearing loss. So it's not used for sensorineural, it's only used for conductive hearing loss. Because what hearing aids do is they're actually magnifying the vibrations, the sound vibrations that are coming through the ear. And remember, that is what's happening with the outer ear, right? So your pinna and your ear canal, the sound waves are traveling through. So if there's something wrong with them and the sound waves aren't traveling through all the way to the middle and inner ear, we use a hearing aid to amplify those sounds so that they can travel through. And hearing aids can be worn in or behind the ear, and there are two types. You've got analog, which are sound waves. So what they do is analog hearing aids convert sound waves into electrical signals, which then and they're amplified, and sound waves are fed back into the ear. But if you've got a digital hearing aid, sound waves are actually converted into numerical codes that then amplified, and sound waves are fed back into the ear. So the difference here is we're going from sound waves to electrical signals in comparison to sound waves to electrical signals in comparison to sound waves to numerical codes. Just please ignore my attempt at drawing a straight line. Let's move on. So how do they work? Here's a more detailed process. So sound energy is converted into electrical energy via a microphone, and the electrical signals are sent to an amplifier, which produces louder sound waves, and then through that a speaker directs the amplified sound waves to the auditory canal, so it goes straight through, and then it travels the normal pathway through the ear to go via the cochlear auditory nerve to the brain. Bone induction implants, these are the ones that are used when there is conductive or mixed hearing loss. So it is an alternative to hearing aids because we place a processor behind the ear, and we are essentially, what happens is sound waves are detected by the processor. We convert those sound waves digitally into vibrations, and vibrations are then transmitted to the implanted section of the device. So inside, in bone induction implants, they actually insert an implant inside your ear, like inside just this part here, in front of the ear, yeah, just this part here. So they put an implant in your inside your ear. So what happens is the processor receives the sound, it converts it into vibrations, and those vibrations are immediately sent to the implant. And the implant, what the implant will do is that it will stimulate vibration of the ear bone. So vibrations are transmitted from the bone to the inner ear, and that is triggering fluid movement in the cochlea, and that will then lead to the electrical impulses, the creation of electrical impulses from the cochlea, so that it can be sent to the auditory nerve via the brain. So this is where we kind of again, we are focusing on the amplifying the sound, sending the thing straight, sending it straight through, and that's why we've got an implant inside the ear as well. Okay, So here's a more detailed process. So transmit a sound from bone to inner ear, bypassing the outer and middle ear. So we're not worrying about the eardrums, the ossicles, or the outer ear. And the sound, so there's a sound processor which will detect the sound waves, it will analyze them, and it will amplify them and convert them into vibrations. Then the implanted part will actually transmit the vibrations from the bone to the ear, and through that the inner ear, the movement stimulates the hair cell, which result in the electrical impulses, and then it goes to the brain via the auditory nerve. Okay, cochlear implants. This is where this is used when there is a sensorineural hearing damage. So you've got hearing damage happening to the inner part of the ear, and because of that, we now are actually using a device which is so electronic device replace the damaged inner ears. It's replacing the function of the cochlea, and the function of the cochlea is to actually convert the sound waves from kinetic into electrochemical impulses so that it can be sent through. So what happens here is we've got a transmitter which is going to receive the sound. So it's going to receive the sound, it is going to turn it into a digital code, and then from there the code will be transferred to internal implants. Again, we've got an implant inside the ear, and that from that implant, what happens is, because we've already converted into a digital code, we're transferring into the internal implant. The code is converted, it is converted into electrical impulses, and then the impulse, after it's been connected, it's going to go straight through via the auditory nerve to the brain. It's surpassing the cochlea. It's going straight from the implant to the auditory nerve. That is what is happening. Okay, I'm going to try and hurry up. I do know we are in time. So that's the summary. I'll leave that for you guys to have a look at. Here is the structure that you need to know. So the eye, I would definitely recommend checking out that YouTube video, because YouTube videos make everything so much easier to visualize, because it actually shows you how light travels through, and obviously I can't do that with my hands. I can try, but it's just not as effective, so check that out. And so essentially what happens is light is entering through the cornea. The pupil lets light into the front of the eye. The lens focuses light by actually either shortening or lengthening based on the light and the direction from the space that you're looking at. And the light rays, they meet at the focal point. So this is the focal point at the retina. So the focal point is here at the back of the eye. That's where the focal point is. And rods or cones are excited by light, so they convert signal into electrical impulses, and then those electrical impulses are sent to the brain via the auditory nerve. Accommodation is it's basically a process where the lenses and cornea help to refract light. So lenses can change shape, but the cornea cannot change shape. And the goal is for the image to land on the retina. It has to land on the retina, and so that it can be interpreted by the brain. But that may not always happen, and this is why. This is a normal eye. This is the focal plane at the back. Here we've got the retina. It's landing right at the back of the retina. That was a disappointing line. Please do not worry about that. Okay, so yeah, there's a line there already. So we've got the normal ear, we've got the focal plane. Now, what happens is, in a myopic eye, the focal plane is too far in front of the retina, and that means that you're nearsighted. You can read or see things that are close to you very clearly, but when it comes to looking at things that are far away, because of your focal plane being so in front of your retina, that can mean the objects that are far away are blurry to you, and that is and it can happen if the cornea is too curved or your eyeball is too long. And hyperopia can happen when your focal plane is too behind, it's too far back from the retina. And what that means is you can perceive things at a distance really easily, it's really clear. However, you have farsightedness. You can't see things that are close to you very clearly. So objects up close can be blurry, and that's kind of the opposites of each other. So this is where so that's more on myopia and hyperopia. So that's that. Now, another disorder is astigmatism, and this can happen when the shape of the cornea is asymmetrical, and that can cause vision to be blurred at all distances. Now, astigmatism can also be genetic. It can also be from birth. So with And eye with astigmatism basically can see things don't land on the focal plane. That is what's happening. They're not landing on the focal plane like in a normal eye. Because of the shape of the cornea, they're landing like at unfocused places. So others include glaucoma, which is the blindness from buildup of pressure in the eye, and it can also be, and that can also cause optic nerve damage, and cataracts. That's clouded areas of lens, blurred or tinted vision. So this is where we use glasses to assist us. So we change angles at which light hits the cornea, and also glasses changes the focal point, restoring normal vision. So if you have myopia, you would wear concave lenses, so that essentially what happens is, because it's concave like this, the image is going to hit further rather than just stopping in the middle there, right? That is what was happening earlier. The focal plane was too far in front of the image, was stopping there. Now we want the image to go the whole way. However, with hyperopia, if you've got convex lenses, so with hyperopia you need convex lenses because the image was hitting too far in front, but we want these curved lenses where the image would then stop hitting, just at the focal point there. So there's a summary. Myopia is nearsightedness. I just like to remember it as MN, like myopia, nearsightedness, and then that helps me remember hyperopia. That would be the opposite, but if you can find like a better mnemonic or something or acronym maybe to help you by, that'd be great. And yeah, so these are the structures of the eye. So strategies and advice, I'll quickly finish off on this. I know we've gone a bit over time. Now remember, your self-worth doesn't depend on your productivity. It is okay to take breaks. It is okay to some days not feel your 100% and smash out everything on your to-do list. It is totally okay. Please take time for yourselves. So types of study, you can make notes. It's good, it's amazing, and it's a good revision activity. But practice tests are the most important methods because you want to use exam conditions. You don't necessarily have to do that every time. You could just do practice questions, but closer to the HSC trials, you do want to take that time to do practice questions and mark them. Look at the marking criteria. Give yourself feedback. Send it to, I know with like my students in my biology class, because I teach biology and English and PD as well here at CHSMA. With me, it's always like, send me your work. I want to mark your work. Send your work to your teachers. Get feedback on it. That's really important. And that's something I used to do as well. Always sending work through. And giving those follow-up emails. Hey miss, did you get a chance to have a look at my work? It's totally okay to do that. Your teachers want to help you out, and they will make the effort if they see that you're really trying and you're really on top of your stuff. Other study methods, flashcards. You can obviously make your own. You can make them online. We have a new range of flashcards here at ATA Notes, and they include biology flashcards, and they are amazing. I've had a look at them. They cover every aspect of the syllabus. They're clear, to the point, really awesome for that active learning. So if you're interested in that, that could be something to check out at our shop. Find and make and print posters. There are apps with bio quizzes that you can use, but they're not always reliable because sometimes the questions may not be you don't need to know them. Watch YouTube videos. That's really important. Like, that's important in the sense that with some of the processes, they can really help you understand. Like with the whole, for example, how light, like the whole eye process, definitely check out that YouTube video. Studying with friends can be a really good source as well. Practice tests, those are some sites where you can find some practice tests as well. Obviously lots of practice tests here at TuteSmart if you want to check us out. Textbooks and websites. YouTube, our Amoeba sisters are great with their animations. Like, it's really easy to understand. Apps and seminars as well. And obviously we've got free lectures on here at ATA Notes, and you can also access past lectures. Pomodoro is a technique to study, like 25 minutes study, and then take short breaks every time, and then after 4 Pomodoros, take a longer break. Noise, what level works for you? Some people like studying with music, some don't. I don't think it's necessarily proven quite helpful to study with music. A study environment is really important, and make it fun. Get some stay sure to motivate you. Yeah, now some tips. So yeah, I generally did 4 to 5, sometimes 3, depending on like the days and the time. Because sometimes I had to like stay back after school to help out at events and stuff because I was helping out. So did 4 to 5 hours of study generally every afternoon, and sometimes maybe more if there's like assignment due or something. Again, weekdays, weekends, try and get 5 to 6 hours done, but you know, you can definitely take time off if you need it. Try to get in at least 2 to 3 hours by study a week. I think that's pretty manageable. And take breaks. Take breaks. That's really important. That's it. Managing time. During reading time, read your questions. Decide how you'll do the exam. I like to just do it front to back. It just helped me like remember everything. But there were times where if I was like, okay, I need to come back to this question, I would do that as well. Yeah, there's the structure of the HSC for you. And finally, checklist for a full mark answer. Now, introductory and closing sentences, judgment statements, key definitions, and examples are really important. You can use diagrams and tables and flowcharts to help you as well. They can be really helpful, and they can help you gain, show a better understanding and gain some more marks as well. Amazing. Now, these are our verbs. This is to help you sort of understand, how you should be structuring your writing. The difference between a compare and a justify or a proposed question is huge. That is it, guys. So definitely check out the slides for some of the bits that I may have just kind of gone through. Obviously, check out some past lectures if you want to have a bit more detail on like the Mod 7 stuff. But thank you so much for tuning in. I really appreciate it. I will see you guys later. Best wishes for your trials. You guys will smash it. Thank you so much for tuning in. Best of luck and take care.